Hello, everyone, and welcome again to an webinar with Ageless Partners here. And today we will have the topic of Ageless Epigenetics. And before I jump in, I'll give a brief overview of Ageless Partners, and then I'll introduce Kamila. So we are a small startup company, and we really have two underlying themes or missions. The first one is to accelerate our progress towards an ageless future. So that means speeding up the development of life-saving technologies and changing the public perception on what aging is and the fact that it is reversible. And the second one is all about educating people and helping them to understand what we can do today to slow down and reverse your aging, as well as what is on the horizon. We have a wide variety of free research on our website, a biohacking platform that's also on our website for free. And we also have coaching sessions that we offer as a health coaching service. And we're also going to be introducing workshops where we'll have a Q&A type format soon in the future as well. So we're really looking to learn more today on actionable insights on what we can do to be healthier, to optimize our health, and to increase our longevity, and to really understand what are these drivers that we can then manipulate, these drivers of aging and disease that we can then manipulate through biohacking, through changing our habits, and through life-saving technologies. This will be our second in a series for epigenetics, and should be interesting. A bit about Kamila. So Kamila Issabayeva, she's an Ageless Researcher here with Ageless Partners. She's currently in graduate school and studying hard, so we really appreciate her putting this information together for everyone and taking the time to piece this together. She's a nutritionist and has studied various areas of biology, computer science, and is now digging deeper into the science of aging. So today, Ageless Epigenetics, part 2. I'll let Kamila take it away. Thanks.
Thank you, Jason, for this brilliant introduction. And today, I'd like to talk about the mechanisms behind the epigenetics in detail, and also what epigenomics changes happen during aging. And also we will talk about some environmental and extrinsic factors that actually influence our epigenome. And we'll start with a quick recap for those who were not with us last time. What is epigenetics? And our epigenome is a collection of chemical tags that affect DNA and its related structures. And basically, any genetic expression modifier is what actually regulates our epigenome. And some of the chemical tags include post-translational histone modifications and DNA methylation, which are key epigenetic processes that regulate our epigenome. And they result mainly in modifying the chromatin structure, which changes its state from open to condensed state and vice versa. And some of the enzymes include histone acetyltransferases and histone deacetylases, for example, that modulate or control gene expression. And actually, our eukaryotic genome, as you can see on the picture, is composed of a basic unit, and basic unit of eukaryotic genome is chromatin. We can see it on the picture here. And chromatin is composed of nucleosomes, which are composed of histones. And histones are basically proteins that are wrapped around our DNA, and histones composed are of 4 units of proteins, such as H2A, H2B, H3, and H4. And they have amino terminal tails, which are histone tails that are exposed on the surface of nucleosomes, which can then be modified, and they are subject to post-translational modifications. And what happens is that different groups attach to those histone tails, making our chromatin change its state. So, for example, to the right, to the left, we can see that closed chromatin is when our nucleosomes and histones are densely packed, and transcription cannot occur due to inaccessibility of DNA to transcription factors. And on the other side, we can see open chromatin, which is usually called euchromatin. Is loosely packed, and transcription can occur because transcription factors can access our DNA. And what molecules modify histones or attach to our histones? They are, as you can see on the picture, are acetyl, methyl, phosphorus, also ubiquitin protein or small ubiquitin-like modifier, and also ribose. And there are some other molecules that are still being discovered and actually also influence our epigenome. However, there is another mechanism, how our epigenome is regulated. Histone modifications also affect transcriptional activity via particular enzymes that recruit transcriptional activators or suppressors. And again, enzymes involve enzymes such as histone acetyltransferases and methyltransferases. They add or remove groups to our histones, and this is another mechanism. And what actually transcription factor does is that they bind to a particular sequence in our DNA, and then they activate or recruit nucleosome remodeling particles, which then remodel our chromatin, and our gene can then be transcribed to finally produce a protein. And also some transcription factors can initiate remodeling by binding to even closed chromatin and mediate nucleosome remodeling and change histone marks. And overall, what we can see in here is overall gene regulation process. So it starts, it happens via stepwise mechanism, and it starts with a pioneer transcription factor binding to our DNA, and then this makes nucleosome remodeling, this recruits nucleosome remodeling particles, which remove or displace actually the nucleosome, and our DNA becomes accessible. And now it means that transcription factor can transcribe the sequence and make our gene or produce the product, the protein, using RNA polymerase and some other transcription factors. However, what happens during aging is that every step can change actually. So for example, some of the changes include loss of heterochromatin, nucleosomes, or repressive marks that can actually remodel our chromatin, and also altered pioneer transcription factor activity and change in transcription factor activity and dysregulated gene expression. And this everything contributes to the nucleosome occupancy, and finally changes in one step can have downstream consequences for gene regulation overall. So that's why it's really important to understand this process. And what we can also see during aging is that we there is loss of histone, and loss of repressive histone marks leads to decrease in nucleosome occupancy, and finally heterochromatin protein binding. And these changes are associated with a loss of nuclear lamina. And also in euchromatin, we can see histone marks changes, which together they with DNA methylation increase its specific loci influence to alter gene expression with age and the progression of aging. What we can see in here on the slide, on the picture, is a comparison between heterochromatin and euchromatin processes happening in young and old cells. And what we can see in here is that there is loss of repressive histone methylation, and also loss of histone binding proteins, and lower nucleosome occupancy happening in our cells. And also there is a global tendency of heterochromatin DNA hypomethylation, whereas there is hypermethylation of euchromatin that change actually our transcription, that actually contributes to the changes in transcription regulation. And overall, what are the trends happening during aging is that it happens that DNA hypermethylation, for example, happens in genes that are responsible for development of anatomical structures such as organs, and also in the regulation of transcription. And genes responsible for nucleotide biosynthesis, metabolism, and control of signal transmissions are differently methylated between centenarians' offspring and offspring of both non-long-lived parents. And also there is a change in DNA methylation and global hypomethylation in brain, whereas there is hypermethylation in genes that are responsible for neural development. And that's why it's epigenomic changes are really important in when considering what we can do to reverse aging. And actually this what also like supports the thought that actually our epigenetics has more influence on our aging patterns. And also epigenomic changes lead to are connected with all other hallmarks of aging, and moreover they can trigger the emergence of other hallmarks as well. And another interesting thing is that our epigenomic patterns are actually heritable. And so here, for example, we can see that there was a study that researched whether offspring of males which fed low protein diet exhibited the same epigenetic patterns as their parents. And what was discovered is that decreased levels of cholesterol esters and some other epigenetic characteristics were passed to the offspring from parents. And that's another important considerations why we need to consider environmental reprogramming in order to provide our children with beneficial epigenetics. So some of the sources of epigenetic variability include psychological state, social interactions, alternative medicine, drugs, microbiome, exercise, drugs, toxic chemicals, disease exposure, and of course diet. And these are all things that we need to consider and that we will talk in detail today. I would like to start with a role of reactive oxygen species in gene regulation. So it is said that it is believed that aging cell is exposed to a reactive oxygen species, and they actually increase inflammation and induce DNA damage. And also they influence DNA methyltransferase activity, which is actually an enzyme that is responsible for our DNA methylation. And the damage results in an increase in mutation frequency and finally affects the DNA methylation. And what actually is interesting is that there are transcription factors that are sensitive to reactive oxygen species, and signaling of reactive oxygen species from mitochondria can impact chromatin-mediated silencing in yeast. And there is also mitochondrial dysfunction, and also mitochondrial dysfunction results in increased methylation of histone 3 lysine 9 residue and global compaction of chromatin. In here, we can see the effects of ROS signaling from mitochondria, and that leads to oxidative stress, cell damage, and low bone mass, cell signaling, altered signal transduction, mitochondrial dysfunctions, which are all observed during aging as well. And another thing is that mitochondrial stress can trigger large-scale changes in heterochromatin, and suggesting a link between age-associated stress response and the formation of heterochromatin, which is generally observed during aging as well. And here are some of the factors that influence mitochondrial dysfunctions. For example, sedentary lifestyle, genetic predisposition, inflammation, and chronic infections, nutrient deficiencies, stress and sleep deprivation, sugar and processed foods, environmental toxins and radiation exposure. And these are all things that we also need to set up, and we need to consider and keep at its best, as to say, to keep our mitochondrial wellness and to keep our mitochondrial health. And another, so what we can actually include in our diets to sustain our epigenome, and it is well established the role of antioxidants as dietary regulators of epigenome. So for example, zinc, selenium, vitamins A, C, and E, some of the non-nutrients such as lycopene, polyphenols, and these are all effective in scavenging free radicals and preventing damage to DNA, proteins, and lipid in the cell. However, there is also emerging specific functions of these things in epigenomic regulation. So, for example, selenium in the form of selenomethionine induces base excision repair and also reduces DNA methylation potential by altering adenylyl homocysteine concentrations, and which actually regulate one carbon methylation pathway. And another specific role of zinc is that it also affects DNA methylation, and specific role is still being discovered, but higher intakes of zinc in older individuals may ensure optimal function of base excision and nucleotide excision repair. And because proteins involved in the repair systems are actually zinc-associated proteins. There is also a discovered positive effect of a kiwi fruit per day. So extra fruit increases the antioxidant status of plasma, and it decreases the endogenous inflammation happening in our cells, but also reduces oxidation of DNA. And in addition, also it stimulates base excision repair, and we need to just consume as little as 1 kiwi fruit per day to keep our DNA damage as minimal as possible. And another important dietary inclusion is folate as a DNA repair regulator. And low folate status limits the cell ability for nucleotide excision repair. And folate and its partners such as vitamin B6 and vitamin B12 are the most important methyl donors, which are actually important for DNA methylation, for example. And some of the sources of folate include leafy greens, asparagus, broccoli, papaya, Brussels sprouts, seeds and nuts, of course, avocado, okra, cauliflower, beets, and bell peppers. And another components that interfere with various epigenetic targets also influence cancer prevention and therapy, include curcumin, the component of soybean, T polyphenols, resveratrol, sulforaphane, and which can be obtained from cruciferous vegetables. Resveratrol can be obtained from grapes, T polyphenols from tea, and soybean and turmeric. Also, polyphenols mediate the reversal of abnormal epigenetic alterations and therefore can change abnormal gene expression. And these components actually mediate the induction of tumor suppressor genes, and while inhibit the tumor promoting genes, such as the human telomerase reverse transcriptase during tumorigenesis, like generation of tumor processes. Another important vitamin to consider is vitamin D, because it is a huge transcription regulator. And vitamin D can be utilized as an epigenetic therapy for cancers at early stage based on its roles in demethylation and upregulation of tumor suppressor genes. And some of the sources, as you all know, include egg yolks, salmon, cereal and oatmeal, soy milk, canned tuna, shrimp, cod liver oil, which is also a great source of vitamin A, and some other fat-soluble vitamins, mushrooms, oysters, and so on. And vitamin A is also a great epigenetic regulator. So also, studies have shown that vitamin A could affect histone methylation of genes that are involved in the production of several cytokines and change the cytokines response, which are really important for inflammation and stress response. And some of the vitamin A sources include liver, fish, carrot juice, cheese, sweet potato, squash, and also cod liver oil, as we discussed before. And vitamin B is also a good epigenetic regulator, and especially biotin, because it is a substrate for histone biotinylation. And biotin is another group that gets attached to histone, and niacin is also involved in ADP ribosylation, which we've discussed before. And some of the sources of vitamin B include oats, spinach, nuts, soy milk, eggs and dairy, beans, chicken, fish, and beef liver. Another important things to consider are chronic inflammation and caloric restriction. They are also great epigenetic regulators. So it is said that chronic inflammation, which shortens lifespan, accelerates methylation drift. And what is methylation drift is actually the process that we observe during aging when certain genes get hypermethylated, whereas another genes get hypermethylated. And caloric restriction said to delay aging-related methylation drift in mouse, in rhesus monkey, in human blood cells and other tissues, and also decreases the methylation age. And here we can see the schematics of how exactly the caloric restriction affects the our epigenome. And actually, apart from this, caloric restriction has been shown to have significant effect on DNA repair pathways. And though caloric restriction extends longevity, the extent to which caloric restriction affects the age-related gene expression may be species-specific. It is important to say. And Chronic moderate caloric restriction can expand lifespan and enhance health of a primate, but it also affects brain gray matter. But however, it does not affect cognitive performances, which is good. And here on the picture, we can see that caloric restriction influence the methylation, DNA methylation by increasing the DNA methyltransferases, and hypermethylation of key regulatory genes such as RAS and P16, which result actually in reversal of aberrant gene expression during aging and maintenance of chromatin stability and extended longevity. And also it increases number of histone deacetylases, which are enzymes that remove acetyl group from histones, and it leads to increased activity of sirtuins such as SIRT1, which is beneficial for longevity generally, and also responsible for DNA repair and some other beneficial processes, and downregulation of P53, such as which is like a really well-known proto-oncogene and tumor suppressor gene, and which also result in extended longevity. There are also extrinsic factors that we need to consider that modulate the aberrant changes of epigenetic pathways. And here we can see the schematics that how exactly the exposure to environmental factors influence our epigenome. Here we can see that exposure to toxic chemicals, for example, leads to oxidative stress and inflammation. It also affects our methyl group availability and also availability of enzymes that are needed for right gene regulation. And also they all influence on our processes such as DNA methylation, histone code, and microRNAs, which are also important regulators of gene transcription. And they lead to chromatin condensation, which actually, as we've discussed previously, lead to buildup of gastrochromatan, and finally can increase the risk of disease. And some of the extrinsic factors that actually had a detrimental effect on our epigenome is tobacco smoke, metal ions, bisphenol A, benzene, alcohol, and many other toxicants. They induce epigenetic alterations. And actually, I would like to talk in detail about each of those in our next presentation. So this is the end of my presentation, and I would like to hear anything, any suggestions, questions, comments if you have, and thank you for your attention. And yeah, that's it.
Thanks, Kamila. No, that was fast, but that was good. That was very detailed. Yeah, I thought that it was definitely insightful.
Thank you.
Yeah. I'll just open it right up to everyone. If everyone has questions for Kamila, I'm sure I have some, but feel free to jump in and ask any questions you may have as well.
Hi, Kamila. Nice presentation. Are you surprised by the findings, in fact?
Well, yes, actually, they were interesting to read about, because when I was preparing, I was like really excited to tell like people, if I'm not sure, like people who are generally interested in like aging, they already know that, but I was actually counting on like people that are interested to know like more about epigenetics, and I was really excited to know myself about that, and also to talk about this in like broader audience. So yeah.
Because in fact, already Hippocrates told that the origin of disease is badly digested food, and that the relents create vapors and they create diseases. So you see, we go back to the roots. I have one question, perhaps. It was nice to see also that that father diet has an influence on offspring, because usually one blames the mother. Now, mice protein diet is probably not equal to human male diet, but was the study set up to see for how long this diet had to be specifically altered, or is it just a point of experiment?
Well, in this particular study, there was not like time restricted details. However, there are another studies that are actually say that it is important, like to, for example, there were some benefits of like dietary restrictions had a potential influence during not in like younger not in younger mice, but in older mice. And this is like one of the important considerations. However, there was also like important that longer, even like short period dietary interventions, they influence our epigenetic patterns really well. And that's why even like short term changes to like feeding regimens, for example, are actually advised because they influence our epigenetic patterns. And yeah, generally, I would say that time actually really matters. Yeah.
Makes sense.
And
Hopefully
yeah,
for mostly wanted pregnancies. Okay,
yeah,
thank you.
I would be happy to provide like more detail how exactly the time influences, and this is something for like my further research. Yeah.
Definitely a good point. And I think you bring up a few good points actually. I mean, just that having that variation. It's like if you ate the same thing every day, you're not really, you're not really varying your diet enough to get all these different benefits from each vitamin, and even just varying how much you fast, how much you restrict your calories. It's definitely, I think there's definitely something here. And I like that you've kind of mapped out, the epigenetic processes that are affected. So one thing I've been looking more into is this gene therapy we came across recently, and it's very similar to a follistatin gene therapy. What are your thoughts, Kamila, just on these like gene therapies, these new technologies that are now able to manipulate genes through a wide variety of techniques? Like how do you see that kind of fitting into this?
Well, I'd say that I'm really excited about this technology, but there is still a lot of like things to know, because we are still yet not like not into. I think that we need to like increase our potential and our knowledge in actual gene functions, and what actually like be the consequences of, for example, turning on or turning off a particular gene. And this is yet something like we need to do in order to greatly benefit from the gene therapy. And however, there are emerging epigenetic drugs like that change actually our epigenomic patterns, and that is something I would say that maybe have like a more recent potential, because like epigenomic patterns are like a little bit easier maybe to change, and in that sense it is more like accessible probably. But yeah, I would need to like a little bit more knowledge into that to say exactly. But this is at this point, this is my view into that.
Totally makes sense. And I mean, any like therapy or drug, obviously there could be some side effects, but but really like people love to eat. So if we can just continue to find therapies that kind of mimic caloric restriction, I feel like those are going to be definitely useful for people, because people they don't mind a little fasting, but people just I find it kind of hard a little bit. So I think any of those, and this follistatin gene therapy, and I'll definitely send you more info on the one that I'm looking into by Mini Circle, and it sounds like they found out a way to even reverse it if necessary. And it sounds like it's because from what I understand, when Liz Parrish did her gene therapy, there was no off switch. And so what this company Mini Circle is saying now is that they do have an off switch, and they've figured out a good delivery mechanism, and they've came out with this technology that they seem to be pretty convinced. The guy who's the founder said that he's injecting himself now. So I think that tells me that they're onto something, and that this could be a life-saving technology. And I definitely, and maybe what we'll do is have another talk just kind of to go in this therapy in more in depth, because I think I want to understand the biology. I want to understand, is this something if they're claiming, hey, this is low risk, well, like there's always risk, there's always some risk, but what are the potential unknown unknowns, and what are the known unknowns too, to some degree, because manipulating genes, I'm sure we don't know it all yet, for sure, right?
I've heard about Mini Circle, and what's interesting is that they use like plasmids to like transfer genes, and I was really surprised by that, because like when I was little, I was like when reading about bacteria and horizontal gene transfer, I was thinking like, wow, this is a really interesting like trait that they have. And now when Mini Circle is doing like the plasmids, like transferring genes through plasmids, I was thinking, well, wow, this is really something like from sci-fi fiction.
And I mean, so like, and I mean, you probably understand this more than I do. I mean, let me share my screen real quick, Uma. I'm just curious about like when you're talking about plasmids, and they do have that on the website. I just want to know like, what are the risks involved?
I'm actually interested to know what Ms. Helena thinks about like the gene therapy technology and some like Mini Circle, this plasmid gene transfer. If you have any like thoughts.
When you say plasmid, it reminds me when I was studying medicine, I wrote an article about this even before I was graduating enough. So that's why when I heard first about plasmids, you said or showed very well that it's complex, and that there is also a reason why some genes at a certain moment get repressed. Because when we have no repression, we get kind of immortalized cells, and what the immortalized cells means, that's usually havoc in an organism. So I would be a little bit cautious, Justin, when you say a therapy, because a life-saving therapy, because then it depends on the target. And as you said, Kamila, or also you, Justin, is you always have a trade-off. So the trade-off has to be really known before you start inject anything. What was nice in your presentation, that you find all these epigenetic modifiers in such a large scale, and that you also find the food that you find everywhere. You don't need to import the kiwis from Chile to Europe. Will be anyway too costly, short, but that it's in the mix. And when you said you get brain gray matter downregulation, but not for the cognitive.
Yeah.
Very nice. You need also the rest of the non-cognitive. So
Yeah, sure.
it's a very subtle, what you showed nicely is that it's a very subtle homeostasis or balance we need to achieve. And the moment you get in, you will get artificially in, probably in a much too strong and unilateral way, and you will have to see what's the trade-off of it.
Yeah, sure. Thank you so much.
Stay with the holistic approach, because that's finally what will probably be the part that will be the best or easiest to integrate also on a large societal scale. And then there will be probably the very specifica when you will not feed the tumored cell with a specific agent mix, but that's another line. Thank you.
Yes. Thank you so much. Appreciate your comments.
Thanks. No, that was well said. And I kind of wonder if we know what some of these trade-offs would be with this gene therapy. Like, I mean, the way I understand, it's producing this additional protein that our body already produces, and when you do this gene therapy, you're enhancing, you're able to produce more of this protein. And I kind of wonder what could be the negative side effects, if any. I don't know if you have any thoughts, but.
You have also already take the diseases we know about, chronic kidney diseases, where you get traditionally, I know it from my family two generations back, when you get a protein sparing diet, because the kidney just can't do with eliminating all this superfluous protein. Example are some of the bone marrow diseases and lymphocytic leukemia and so on, where you get all these huge amounts of protein that's generated and then has to get eliminated. So it destroys, in fact, your renal function. So you see, either it gets deposited somewhere where it has nothing to do, or then it gets eliminated and it's competing with other functions.
That's a very good point. That's very good. That's a good point.
It doesn't get all in the muscles, so sorry guys. So
right, it would have to be filtered out.
Around.
So it could affect your kidneys, but because it has to get filtered out somehow. Yeah, that totally makes sense. Huh. That's a good point. That's a very good point. Kamila, any additional thoughts on that?
No, I totally agree, actually. Yeah, that's something I never thought about, so I appreciate it.
For sure. Yeah, it's something to definitely think about, and even the magnitude, if I'm an older person and I'm at risk of becoming frail, sarcopenia, osteoporosis, that may be a bigger risk than the kidney risk. But of course, it's all relative. It depends, probably. I mean, if you have kidney problems already, maybe not. Maybe that's a factor that should be taken into account. So definitely a good point.
But you're right with the geriatric frailty, and there comes just the problem to eat a well-balanced diet, because the gustatory effect somehow vanishes. It doesn't taste the same when we were young. Well, it's also because the fruits are picked when they are not ripe enough and so on. But when you are really in geriatrics, it's a problem to get people to eat reasonable amounts of a mix, because it doesn't taste the same, because you can't chew it the same, and And so you have to counterbalance it with colors and all these tricks you hear or you see when you go around in elderly care. But that's a real challenge, and probably maybe even the studies you're doing, Kamila, will go in this direction, how to get the optimal mix in once where you're farther away from it, you're 60, 70, 80, or even 90. Yeah? That's clear. It's not all the old, very old Japanese women on this southern island there, because they are probably genetically trained already since ever to have their specific fish, soja, and whatever diet. Sorry, but I'm monopolizing the discussion.
No, no worries. You're good.
Yeah, this is good. I love how our discussion goes, and yeah, this is something for for our consideration.
Yeah, it's a tricky problem for sure. Thanks for your perspective, Helena. Anyone else have any thoughts on any of the slides or topics?
Yeah, Phil here. Yeah, thanks for an interesting presentation, Kamila. It was really good. The thought occurs to me, though, that a lot of this stuff is sort of fairly well known, and you reinforced a lot of that. But I guess the problem that I have is that it's one thing to know what you should be doing, and another thing to actually be able to get or keep to that sort of regime. For example, I'm on Sydney time at the moment, and I should be asleep, but it's the middle of the night here, and I thought this was an interesting thing to watch, so I'm watching it. But that's upset my sleep pattern, although I have a terrible sleep pattern anyway. The other thing is that with what's going on with your epigenetics, the other thing is that if you're tending towards a vegan diet like I am, then it makes it even harder to keep to what you should be doing, in quotes. And the other thing is that if you are interested and serious about this sort of stuff, then you need some sort of mechanism of measuring what your current state is at the moment. And that's why I'm interested in epigenetic clocks and other mechanisms that can say, this is what your current state is like, and these are the things that you might do to try and rebalance or fix those problems that are shown up by various measurements or whatever. But yeah, it was an interesting presentation. Thanks for that.
Thank you so much. I hope you get enough rest and sleep, because yes, getting enough sleep is crucial, and actually circadian rhythms are also important epigenetic regulator.
No, well said, well said. And it's a good point too, Philip. Like, I think it is one thing to know, but actually doing some of this stuff in practice is incredibly hard. And ideally, like, the lifestyle hacks can get us somewhere. But the goal is to really have more powerful and more robust therapies that when we slip up a little bit on our lifestyle, that they'll have a stronger effect than any small manipulation we could do related to lifestyle. But and to your second point too, I think having that feedback is critical, because a lot of people, they're kind of guessing at this point. If I knew, like, okay, when I live my lifestyle, my epigenetic measurement, let's just call it, right, some number was at a certain level, and I did a bunch of things, and now I can quantify and say, okay, now it's better. That definitely would be kind of a positive feedback loop for me. I think for other people, we do have these, like you've alluded to, these epigenetic aging clocks. So there is a way to do that for sure. But I think having more of a daily or a real-time feedback, that would be phenomenal. I'm not sure what kind of diagnostic tool that would be per se, but that would be something that would be very useful. Yeah. Awesome. Well, thanks, guys. Really appreciate it. Any other final thoughts? I'm sure we'll have maybe around part 3 in the future. We'll see. But I think maybe we'll expand a bit on gene therapies next time a little bit, and kind of compare and contrast some of these mechanisms and procedures, and think about where should we be putting our attention over the next few years. Cool.
Thank you everyone for your time, for your attention, and yeah, we will be focusing also on more like actionable insights into the presentations further. So stay tuned. Yeah, thank you.
Thank you.
Thanks, guys. Appreciate it. Cheers. Have a good afternoon or evening for some people.
Yeah.
All right. Take care, guys.